In this video, I'm going to talk about the structure of the sarcomeres, which are the building unit of the muscles, and then after that discuss the difference in the mechanism of the contraction of the skeletal muscles versus the smooth muscles. So here you can see that there is a sarcomere that runs from one Z line to the next Z line. So this entire length that runs from one Z line to the next Z line is the length of one sarcomere. And then there are other sarcomeres that are attached to this middle one shown right here. Now there are two types of filaments. One is the thin filaments, which are shown in black and are made of actin. And then there are also thick filaments that are shown in red and are made of myosin. Now the points that the actin filaments attach to is referred to as Z-line, and for that the reason this name is given is that the attached one is like a zigzag pattern, and so the name Z-line is given to it. And then for the thick filaments, which are made of a myosin, the discs that they attach to is referred to as M-line. Now there are other structures that you will have to be familiar with, and these are called I-band, H-band, as well as A-band. So I-band is the zone of thin filaments or actin filaments that are not superimposed by the thick filaments. So you can see that this entire length is only made of the actin filaments and is referred to as I-band. And on electron microscopy, you can see that this white region is referring to the I-band. And then there is also H-band, which is the zone of the thick filaments that are not being superimposed by the thin filaments. So here is a length of a 1 H-band. And then finally we have another I-band on the other side. And then the last band is the A-band, which represents the length of an entire thick filament. So this entire thick filament is referred to as A-band. And the length of A-band is always constant, versus I-band and H-band, which will decrease in length upon muscle contraction. So in the next image, I show you that there is a muscle contraction, as a consequence of which the myosin chains will become close to the Z-line, while the actin filaments will become close to the M-line. And so via this process, the entire length of a sarcomere decreases, the length between the two Z-line decreases, and the muscle contracts. You should also note that since the myosin is becoming close to the Z-line, also the length of the I-band decreases, and so does the length of the H-band. But then the length of A-band, which represents the entire length of single thick filament, is always constant. Alright, next I would like to discuss the mechanism of contraction of the skeletal muscles, and then after that discuss the difference between the mechanism of contraction of skeletal muscles to the smooth muscles. So here we have a skeletal muscle, and the membrane of the skeletal muscle is shown in red, and then we have the sarcoplasmic reticulum, the membrane of which is shown in blue. So upon depolarization of a skeletal muscle via the motor neurons at the neuromuscular junction, there would be activation of the acetylcholine receptors. So acetylcholine will be released from motor neurons, and acetylcholine receptors will be activated, which will induce the influx of sodium. And so once sodium goes inside the muscle cell, it will cause depolarization of a membrane. And then there are T-tubules, which are membrane structures that run deep inside the muscles. So via the T-tubules that run deep inside the muscles, therefore the dihydropyridine receptors, which are shown in blue, can get activated due to the depolarization of a membrane, and dihydropyridine upon activation will change conformation and activate the ryanodine receptors that are localized on the sarcoplasmic reticulum. So once the ryanodine receptor is activated, calcium will be released from the sarcoplasmic reticulum. So that's the mechanism through which the skeletal muscles will release calcium from the sarcoplasmic reticulum. And so here we have a structure of an actin, and then another structure of a myosin shown in red. So once calcium is released from the sarcoplasmic reticulum, it will act on the tropomyosin, which is shown as a red line here, to displace it from the yellow spots that are shown right there. So those yellow spots are the myosin binding sites on the actin filaments. So normally myosin cannot bind to the actin on the yellow spots because the tropomyosin or the red line has covered it. Upon release of the calcium, tropomyosin will be displaced on the actin filament, and thus the yellow spots, which represents the myosin binding sites on the actin, will be exposed. So at this stage, the myosin head can bind to the actin filaments. And after that, there would be a power stroke where the myosin head bends and it will cause pulling of an actin filament. In the next step, ADP will be released and ATP will bind to the myosin head, after which the myosin will now release the actin. So here there is a release of myosin from the actin. And then finally, in the last stage, ATP will be hydrolyzed and the energy of the ATP is used to recharge the head of the myosin. And then you can see that in later stages, this energy can be used for a power stroke. And so the cycle continues. So let me repeat that again. Calcium will be released. It will displace the tropomyosin on the actin filaments, causing the exposure of the myosin binding site on the actin. So at this stage now, myosin head can bind to the actin. There is a power stroke where the actin filament will be pulled. After that, ADP will be released and ATP will bind to the myosin head, causing release of the actin from the myosin. And then finally, ATP gets hydrolyzed, causing recharge of the myosin head, and then the cycle continues again. Alright, now next I would like to go over the mechanism of the activation of smooth muscles. So as we just discussed, ryanodine receptors are the receptors that will induce the release of the calcium from the sarcoplasmic reticulum in the skeletal muscles. And the way it worked was that there was depolarization of a membrane of the muscles via the motor neurons, and then there were T-tubules that run deep into the cells and cause activation of the dihydropyridine receptors that causes later on activation of ryanodine that releases calcium. With smooth muscles, on the other hand, there is a different mechanism, and it's that upon depolarization of a membrane, calcium will be released from the extracellular space. And so calcium will flux in from the extracellular space. And then there is also another mechanism through which alpha-1 sympathetic receptors will act on the IP3 secondary messengers to induce the release of the calcium from sarcoplasmic reticulum. So once again, skeletal muscles release calcium from sarcoplasmic reticulum via ryanodine receptors, while smooth muscles have two different mechanisms. One, via depolarization, causes influx of the calcium from the extracellular space, while via the activation of the alpha-1 sympathetic receptors, it will act on the IP3 secondary messengers that causes release of the calcium from the sarcoplasmic reticulum. So this entire mechanism, the difference between the skeletal muscles and smooth muscles, explain why dihydropyridine calcium channel blockers only stopping the smooth muscles, but then the skeletal muscles remain active. So I'm going to ask you this question. Why is it that dihydropyridine calcium channel blockers cause relaxation of the smooth muscles, and thus this medication is used, for instance, for controlling the blood pressure, but it doesn't affect the skeletal muscles? And the reason for that is that these calcium channel blockers will inhibit the influx of the calcium from voltage-gated L-type calcium channels that are on the extracellular space. So it will inhibit the voltage-gated channels that are shown right here by the calcium channel blockers. And since this voltage-gated channels has nothing to do with the skeletal muscles, therefore it will only affect the smooth muscles, but then the contraction of the skeletal muscle will not be affected. Next, I would like to discuss, after the release of the calcium, how the smooth muscles will get contracted. So as we discussed, there is two mechanisms through which calcium can increase in the intracellular space of the smooth muscles, and it's that one, via the depolarization, it can be influxed from the extracellular space, or two, via the activation of the alpha-1 sympathetic receptors, sarcoplasmic reticulum can release the calcium. So once calcium is released in the smooth muscles, a calcium-calmodulin complex will be formed, and calcium-calmodulin will cause activation of a myosin light chain kinase. So myosin light chain kinase will now phosphorylate the head of the myosin, which will cause binding of a myosin to the actin. And then at this stage there would be contraction. And so once contraction is done, then the phosphatase enzyme will remove the phosphate, after which myosin head will now release the actin. So let's repeat that again. Depolarization causes influx of the calcium from extracellular space, while activation of alpha sympathetic receptors causes influx of a calcium from the sarcoplasmic reticulum. A calcium calmodulin complex is formed, which will activate the myosin light chain kinase. Myosin light chain kinase causes phosphorylation of a myosin head, after which, due to the phosphorylation, myosin head can now bind to the actin and cause contraction. And once contraction is done, phosphatase enzyme will remove the phosphate, after which the myosin head will release the actin. And that concludes the discussion of a sarcomere structure, as well as the difference between the mechanism of contraction of the smooth muscles versus the skeletal muscles.